So the unit this morning is on acute viral infections, and we're going to cover influenza, SARS-CoV-2, and a bit of RSV at the end. And as yesterday, if you have questions in the middle, please feel free to unmute yourself and just ask, or put them in the chat. I will watch the Zoom chat. It'll be harder for me to watch the Slack chat, so use one of those two preferentially. Okay, so we're going to start out by talking about influenza virus as sort of our model acute RNA virus. And influenza A is what we call a negative sense, meaning that the genome of the virus itself doesn't actually code for proteins directly. It has to be reverse translated into a positive sense RNA that will actually be the coding RNA for the viral proteins. It's in the family Orthomyxoviridae, and it only has 8 gene segments. And so you can sort of see that here in this little schematic. So 8 segments of RNA that encode the entire genome of influenza virus, and they encode approximately 11 proteins. It varies a little bit by strain. And so the way you get 11 proteins off of 8 gene segments is that there's alternate reading frames that encode multiple proteins. There are 2 non-structural proteins, meaning that those proteins are not part of the virus that gets packaged and sent outside the cell. They're only made inside an infected cell to facilitate the infection, NS1 and PP1F2. And the key proteins that distinguish the different types of influenza virus are its 2 surface proteins, which are the hemagglutinin, shown here in purple, which we also refer to as the HA, and the neuraminidase here as these little spikes in yellow, which we refer to as the NA. And going back to the discovery of influenza virus, the different types and strains of influenza viruses have been defined serologically. And by that, I mean we immunize animals or infect animals with different viruses, and we get antibodies out of those animals from their plasma, from their serum. And if you infect 5 animals with 5 different viruses, and 2 of those animals' antibodies see each other's virus that we do in a binding assay, then we say those are the same serotype. And if the 3 other ones are different, then those are different serotypes. And so it's a very functionally defined assay that does actually relate, though, to the sequence of both the HA and the NA, and tells us a little bit about the evolutionary history of those viruses. So there are, there's actually 18 now serotypes of HA. The last 2 are extraordinarily rare and only found in bats, and there's some disagreement over whether or not we should call them distinct serotypes. But certainly 16 classical lineages of influenza defined by the serotypes of the HA, all found in aquatic waterfowl. And so that's the natural host of influenza is in aquatic waterfowl, and it's generally assumed that all serotypes are circulating at all times in ducks and other aquatic waterfowl, and almost an evolutionary homeostasis to some extent. A subset of those serotypes have been known to infect humans and pigs and other animals. And there will be evolutionary tracks into these different species that will prevent the virus from going back into waterfowl, and the virus stays circulating within a particular group. Similarly, we define neuraminidase serologically. There are 9 neuraminidase subtypes, only 2 of which have been shown to infect humans, 3 in pigs, and other animals for some of the other lineages. So that we define an influenza virus is based on the serotype of the HA and the NA, and that tells us a lot immediately about the range of animals that it can infect. So you'll hear H1N1, and that just refers to those 2 serotypes. As I've mentioned, wild waterfowl ducks in particular are the native host of all influenza viruses, and they sort of form this central role in the evolutionary distribution of these viruses. And then viruses will jump and become restricted within host ranges that can then pass amongst each other if they're very similar. So Humans and pigs often pass viruses back and forth to each other, and you'll see a lot of the same strains in between them indicated on this graph. Whereas chickens, which are avian, like wild waterfowl, have a different set of lineages. And actually, even though they are both birds, they are pretty different biologically. And so the chicken strains often aren't able to go back into a wild waterfowl. But many, many other species have their own influenza viruses, whales, seals. Actually, seal flu is a big problem for certain seal populations. And then horses and dogs also have their own lineages. Okay, so you'll see the viral replication cycle of all three viruses we talk about today are actually pretty similar. All three viruses that we're going to be talking about are what we call enveloped viruses, meaning that they have a membrane around them that's stolen from the cell membrane that they replicate in. So it's a membrane just like the membrane that's on your cell surface. That is where the HA and the NA are encoded. So the virus, in the case of influenza, binds to sialic acids, which are carbohydrate residues that modify many, many, many proteins on the surface of your cells. And the HA protein will bind to those sialic acid residues, and it will trigger the cell to endocytose the virus inside a vesicle. So now you've got the viral genome is encoded in a protein capsid within the virus that's then itself encoded within a viral membrane, which is now inside an endosome. And influenza virus wants to do all its replication inside your cell's nucleus. So it has to get all of that cargo out of this multi-layered packaging here into the cell nucleus. And it does that by the HA protein actually fusing with the endosome, and we'll see that on the next slide, and dumping its genetic cargo through all of these membranes out into the cell cytoplasm, and then quickly shuttling all of those viral ribonucleoproteins, so the 8 gene segments coated with these replication proteins into the cell nucleus, where it will start undergoing replication. And so these negative sense RNA VRNPs have to then be flipped into positive sense RNA, which it does with an RNA-dependent RNA polymerase. So the virus's own proteins, using nucleotides and other material from your cell, take that negative strand, turn it into a positive strand, and then start making more viral proteins using your cellular machinery. And so that's all those proteins are going to be made in the same machinery that makes your own cell's proteins. And then in parallel, the viral polymerases will start making new viral genomes. So they'll take that positive strand and remake a negative strand RNA, which will get packaged with those viral proteins. And all of the viral proteins the cell's making, plus the viral genomes, will get trafficked to the cell surface, where they'll collect and get packaged and then secreted out as new viruses. And to get the virus off the cell surface, because it naturally is going to bind back onto the cell using the HA and the sialic acid residues, the neuraminidase cleaves sialic residues off the cell surface, and that's what actually releases the virus. So that is the full life cycle of influenza. It's fast. It can do this within 6 to 8 hours of infecting, and it's exponential. So each one virus infected cell produces hundreds to thousands to millions actually of progeny virions. So I mentioned that the HA is really critical here. It binds to the sialic acid on the cell surface, and it makes flu have a broad promiscuity in terms of which cells and which proteins it can infect. And because it's really just looking for this carbohydrate modification of proteins, which is really pretty common. And then once it's inside that endosome, this is, the primary site for flu replication is going to be an epithelial cell in your lung where you breathed in the virus. And epithelial cells are not professional immune cells, but they are going to try to not get infected with things. And so one of the things that they do to try to not get infected with things is, as soon as they take in cargo like that, is they acidify the cargo compartment. And so they pump a bunch of acid in there to try to kill anything that would be in there. And this would work pretty well against a lot of bacteria. But flu's actually evolved that when it senses that the cargo is becoming acidified, HA changes its conformation. So it's a chemical reaction that basically unzips the HA protein, and you can sort of see here it's folded up in on itself. This is all these white bands here. It will, under acidic conditions, flip up and stab itself into the cellular membrane of the endosome, that package that's coming in, and then tear open that membrane. And that's what's going to allow the viral genomes to come out. So that's this step right here. It's mediated again by HA. So the receptor that gets into the cell then rips apart the cell membrane that brought the virus in and dumps out the cargo. This is actually the key to why flu in humans and in other mammals is a respiratory disease, because for HA to achieve this sort of conformational switch, it has to have a modification where a little bit of it gets cleaved in a particular way while the HA is forming. And the only enzymes in mammals that do that happen to be localized to the respiratory epithelium. It's sort of it's sort of almost a coincidence. Flu can infect virtually any cell in your body. It's just the new progeny virions are no longer infectious because they can't be modified to make that HA correctly to function. In ducks, that enzyme is present in the gut, and so in ducks, the virus is often a digestive disease. As I mentioned, neuraminidase acts on the other end of the viral life cycle to cleave the sialic acids, which are represented here with these little orange diagrams, off the cell surface so that the virus doesn't stay stuck on the cell by binding back to the cell it just came out of with HA, because you don't want to reinfect the cell you just came out of because that cell's already infected and making virus. And so influenza virus has to balance the activity of the HA, the ability of the HA to bind sialic acid, which is shown here, and the ability of the NA to cleave sialic acid. If any one of them is too strong, if the HA is much stronger than the NA, then the virus is going to stay stuck on the cell and it's not going to spread throughout the lung or spread into your cough. But if the NA is too strong, as soon as the virus attaches to the cell, the NA will cleave it right back off, and you'll never get productive infection. And so there's actually some nice modeling around this balance of the HA and NA, and you can see that evolution happening across different strains. And Andreas has actually done some of that modeling himself. Okay, so what are the key mechanisms on the immune side that protect us from influenza? And this is, again, really a great model of all the acute viral infections we're going to see today. And you're going to see that the immune mechanisms of protection are basically the same, but the manifestations of pathology due to small differences in the biology of these viruses are really quite different. So for all three viruses today, the two primary mechanisms of protection are antibody-mediated immunity and cell-mediated immunity. And both of these, I would say, fall under that broad category of type 1 mediated immunity I mentioned briefly yesterday. And type 1 mediated immunity is that immune class of effector mechanisms that's targeted at intracellular pathogens where the pathogen's replicating inside a cell, and part of our control of that pathogen is going to be to kill the cells that are hosting the pathogen. So antibody-mediated immunity exerts the most pressure on the virus, and this is what leads to actually the rapid evolution of influenza virus from season to season. So the antibodies are depicted here in green. The HA is depicted as these little lollipops, and what the antibody is going to do is, if you've been given the flu vaccine and you've got good antibodies, the antibody is going to block the active site of the HA to bind to your cell, and so the virus can never get inside the cell. And if this works well enough, we call this sterilizing immunity, meaning you actually never get infected even though the virus is in your lungs, because it never gets inside a cell and starts its life cycle. The HA-specific antibodies are the most important because that's the protein that gets the virus inside the cell. You could also make NA-specific antibodies, but it's thought that these don't actually prevent new infection because the NA is not really important for getting the virus inside the cell, but it is important for getting the virus off the cell. So if you block NA, you might have a round of replication, but you might then limit the ability of that virus to spread throughout your lungs. If you've started getting infected cells, you've failed sterilizing immunity, then you're going to need cell-mediated immunity eventually to get rid of the virus. And that's going to be mediated primarily by our killer T cells, the CD8 T cells, which, remember, are going to see antigens in the context of this presentation molecule, this little box on the cell surface called MHC class 1, and it's going to see small peptides from the influenza virus that this T cell receptor on the CD8 T cell happened to be specific for, and they're just going to work through your lungs, finding all of the infected epithelial cells and killing them. An important distinction here is that the proteins that CD8 T cells can see can come from any part of the virus because they're being processed and presented from inside the cell. So any part of the viral protein replication cycle can be processed and presented and have a CD8 T cell corresponding to it. And this is going to be true for all the viruses we talk about. Whereas antibodies can really only look at proteins that are on the surface of the virus. If you make an antibody response to something inside the virus, the antibodies have no access to that, and so they're useless. This is important because the internal proteins of the virus, the parts of the replication process, the polymerases, generally are very conserved and aren't able to be mutated by the virus that much because they have really important and highly complicated functions. And so the virus is just sort of more constrained in its ability to mutate those. We do sometimes see mutation of epitopes, which is this peptide MHC combination, over time in populations in humans that would suggest that there is immune pressure being exerted on these proteins, but it's at a much slower scale than we see the pressure on the HA and the NA. If you think about the kinetics of these responses, which have been measured really to high resolution in animal models and then to reasonable resolution even in humans and challenge studies and natural infection, if you've never seen a particular virus before, you can see it grow up very fast here in red, peaking sort of 3 to 4 days after infection, and then start to initially decline. And this initial decline is not mediated by adaptive immunity. It's mediated by your innate immune response, that type 1 interferon response that your epithelial cells are making on their own. And then your professional innate immune cells are helping make the neutrophils, the macrophages that are coming in the absence of any specific response yet, but trying to alert the lung stromal tissue that there's a viral infection and to try to limit viral replication through these relatively nonspecific mechanisms. 5 to 6 days after the infection is when you start seeing T cells enter the lung for the first time, if it's a totally naive response, if like many of us were for SARS-CoV-2. And they'll grow up to very high levels. And this is what's going to really drive the level of virus down and eliminate it completely. It's also what's going to cause a lot of the pathology and make you feel very sick. So it's kind of why, you might initially feel a little bit bad when you get infected, and then you feel really bad. And that's your immune response really going to town on the virus and then finally clearing it. And during that time, your antibody response is coming up. The antibody response is going to help clear ambient virus in the lung. And it's also going to provide that memory response so that you don't ever get infected with that same virus again, and potentially provide sterilizing immunity against that virus. In a secondary response, all of these kinetics should be much faster. So the CD8 T cell response is now a memory response. It will come up much faster. Your antibody response, if it's an unrelated serotype, is not going to be effective. And so you're really going to rely on your CD8 T cells to respond very fast. If you do get challenged with exactly the same virus, that's usually almost sterilizing immunity. And so when we think about how influenza evolves, we think about two major mechanisms. This middle one is the most common one, which we call antigenic drift, which is we make antibodies to this year's flu, and then that puts a lot of pressure on the HA to make some changes, and relatively small numbers of changes, sometimes just 2 changes, can actually almost fully escape the antibody response we made last year. And so then we have a virus that we're basically serologically naive to. You can think of this as exactly the same thing that's happening with the SARS-CoV-2 variants. It's just happening on a little bit faster timescale than we see with flu, because we've been living with flu much longer, and our immune pressure, I think, is a bit more effective. Flu has an advantage over viruses like coronavirus because it has this segmented genome, and that allows a different kind of evolution that we call antigenic shift. An antigenic shift is when you have 2 viruses from really distinct lineages infecting the same cell in a pig or potentially in a human, and they swap some of these gene segments. And so you end up with this Frankenstein virus that your immune system's never seen before, and could actually represent a complete serological shift. So that it's not even just this small drift where there was a couple amino acids that escaped our antibody response, but a total shift of the HA to a completely different type, which is going to leave your immune system particularly vulnerable and basically treat it as a whole new virus. So everything we're seeing with coronavirus, which we'll talk about, is processive drift. Whereas with flu, we see these occasional shifts, and those are often what drive the pandemics. And if we look at the history of influenza pandemics, the first one that we're really sure of is this 1918 pandemic. And that was H1N1. And it's that because that was when we discovered flu and started naming the serotypes. And in 1957, flu was completely replaced by, H1N1 was completely replaced by an antigenic shift event that was assumed to occur in a pig that then shifted all of the human viruses to H2N2. And that lasted till 1968, when another antigenic shift event occurred that switched all of the human viruses to H3N2. And then in 1978, this H1N1 from 1957 reappeared in the human population. And this is almost certainly the result of a lab release. And this is known because the sequence of this 1978 virus is identical to the last sequences of the 1957 virus. And if it had been circulating in the human population or in an animal population in this window, there would have certainly been drift. And so the thought is that there was experiments in either Russia or China to look at some sort of vaccination study or weaponized study and caused a release. And we had that H1N1 with us in parallel with H3N2 till 2009. And then in 2009, this H1N1 was replaced by a new H1N1, completely replaced. And that's a pretty interesting story as to how that happened. And I'm going to spend a few minutes talking about this in more detail, because we've done some modeling of this in terms of its relationship with the immune response. And that'll be a lot of what the talk's about today. And so the current human influenza strains, both the H3 and the H1, are sort of offspring of that 1918 pandemic. And so that's represented here. This was sometimes called the Spanish flu, single largest pandemic event of the last 200 years in terms of deaths. After that 1918 flu, that virus entered into swine populations, so pig populations in America and Central and South America, North America, Central and South America, and in Europe. And those lineages really diverged from each other because those pigs don't get to hang out together very often. And so we had these two parallel lines of classical H1N1 swine lineages. We had that whole strange series of events that I mentioned earlier, where the 1918 virus disappeared in 1957 and then reemerged in 1978 as exactly the same virus that had disappeared in 1957. And everything was pretty stable for 60, 70, 80 years. And then in the late 90s, this North American swine lineage virus underwent this massive antigenic shift event and started causing this rapid evolution. So it had been evolutionary stable for about 70 years. And then this, what we call the triple reassortant virus, emerged. And this triple reassortant virus was actually an H1N2 and contained elements of swine lineages, human lineages, and avian lineages. And this all happened within the space of about 2 years. So prior to this event, all swine viruses in the Americas were H1N1. After this event, they started rapidly evolving, and they started taking on many, many different human serotypes. They became H3N2, they became H2N1, H1N2, H3N1. It was crazy. It was this massive evolutionary emergence in pigs, and it did cause a lot of concern that something was coming. And so this triple reassortant swine virus hung out for about 10 years. And then this virus met this Eurasian virus, which was still H1N1, had been evolutionarily stable for years. And so this triple reassortant virus met this Eurasian virus in a pig somewhere in the Americas, still controversial as to where it happened. And it donated, through that antigenic shift mechanism, the neuraminidase and the M gene, which is a structural protein, forms the capsid of the virus, and onto this triple reassortant background, and that became the pandemic H1N1. That was the 2009 H1N1, and it's the H1N1 that's still with us. It completely eliminated this old seasonal H1N1, which was from 1978 and from 1957 and really from 1918, but had evolved and drifted for many, many, many years and was now very different from the 1918 virus. And then we also still kept our H3N2 virus. This virus, this pandemic H1N1, many of you might remember, caused a lot of panic. There was, declaration of a pandemic relatively quickly. And then there was sort of not a lot of deaths as a result of it. And we're going to talk about why that happened. But that led people to think that maybe it was a little bit of a bust of a virus. And the truth is it was actually an extraordinarily successful virus from the virus's perspective. It infected about a third of the world in 6 months. And so in that sense, actually much more effective than SARS-CoV-2. It just didn't kill a lot of people. And the reason why is actually buried in this evolutionary history of why it didn't kill a lot of people. Hold on one second. I just saw that there was a question there. We said, why are all RNA viruses not positive sense if they have to be converted otherwise? What's the evolutionary advantage of the negative sense? Andreas gives a very good answer, which is that there are some evolutionary advantages and ecological niches that allow that negative sense RNA to actually offer an advantage, particularly in terms of allowing that viral evolution and maximizing sort of the efficiency of the viral RNA polymerases. And so these actually most of our human pathogens that are viruses are negative sense viruses. They're incredibly efficient for various reasons in terms of the details of the replication cycle. Okay, so this is the spread as of June 2009 of a virus that had infected maybe only a handful of people by March of 2009. So, virus emerged sort of in Central South America and California in February, March, and by June, a third of the world was probably infected. And so why did people not get very sick? So if we think about normal flu pathogenesis, we talk about this U-shaped curve of who's really affected by flu. So the very young infants, children under 2, can get very, very sick and die of influenza, and the elderly can die of influenza. In 1918, we actually had what we called this U-shaped curve W-shaped curve, where those same 2 populations had a problem, but the actually sort of young, healthy adults also were very, 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 very sick. And there was a theory that this group maybe also would have been very, very sick, sort of middle-aged people would have been very, very sick, but that they were protected from some historical H1N1 infection. And that's kind of represented here, where there was a theory that there was an H1 in the late 1800s that protected these middle-aged people in the early 1900s. But what actually happened in 2009 was something very similar. And it's that we had this genetic shift event, but it was surprising because it was an antigenic shift between 2 swine viruses. And the theory before, we wrote this review before 2009 came out, was that one of the viruses in any antigenic shift had to be a human virus. And so in contrast, in 2009, we had these 2 pig viruses that had been circulating with humans for a very, very long time. And all of a sudden created a new virus. And this created a ton of panic because the thought was nobody has protection to this, and it's going to cause 100,000 deaths in the US alone in the first couple of months. So again, that didn't happen. And the reason was because an important group of people, the elderly, had pre-existing cross-reactive immunity to the 2009 H1N1. And you can see that here in, this is looking at antibody titers in people prior to vaccination with 2009 H1N1 vaccine, and looking at their level of vaccination, their level of antibody titer. And so higher numbers here mean higher titers, and 40 is generally considered a protective level. And so nobody here is protected prior to vaccination. But if you look at adults that are older than 60, and you look at their responsiveness to the pandemic H1N1, and this first column is their pre-vaccination titers, they're all over that level of 40. And so this was before they had seen any vaccine. Older adults already had antibodies to the 2009 H1N1. And the reason for that was because the HA protein in 2009 H1N1 came from this North American classical swine lineage that formed that triple reassortant lineage that eventually formed the pandemic. And this North American classical swine lineage entered into pigs in 1918 and then was basically not under any evolutionary pressure because we eat the pigs really fast and they don't do antigenic drift because they're not getting vaccinated and passing change from season to season. So this HA was basically the 1918 HA. And so if you were alive from 1918 to say 1957, then you had seen that HA before and it had just been hiding out in pigs all those years to only pop up again in 2009. If you were born after 1957, then you had no protection. And that was what was seen, is that there was literally lower levels of mortality among the elderly than in a normal seasonal flu season because they had this long ago sterilizing immunity provided to them from their exposure to the virus as a young person. Whereas actually younger people were significantly higher affected than they were in a normal flu season. The risk ratio was really quite high. It was still not as high as the predictions had feared, but it was higher than the elderly. And so it was almost back to that W-shaped curve of 1918. Okay, so we don't have a ton of time left. I'm just going to go through quickly how we've tried to model why these two swine viruses became so effective in 2009 and why their combination was so effective in infecting humans. And so to test this, we looked in human epithelial cells in the absence of an adaptive immune response, just to try to determine what was efficient about the combination of these two viruses. And to do that, we used representative viruses of the classical swine in America, which we call North Carolina, the classical swine in Italy or in Europe, which we call the Italy virus, representatives of the pandemic strains, and a representative of the old seasonal strain. And we measured many features of viral replication and innate immune induction in vitro and primary human epithelial cells. And so first we measured virus growth, and we actually didn't see really significant differences in virus growth, though there was a bit of a lag in the American swine lineage virus versus all of the other viruses that we were looking at. But by 24 hours, actually all of the viruses were almost identical. And in swine primary epithelial cells, all of the viruses were identical except for the human seasonal virus, which is called Brisbane here. This was the virus that disappeared in 2009. So we had a hint that there were some differences, but nothing super dramatic in terms of growth efficiency. And this is measuring infectious virus titers, so the amount of virus that's coming off the cells that are infected. And this is what's quite commonly done in flu studies or in SARS-CoV-2 studies, and it's what you have to model most of the time. At the same time, we decided to measure actually the number of infected cells. And here we were able to see some really major differences, which were obscured by that measurement of productive viral replication. So here we're looking at the number of viral infected cells here in green, and two pandemic strain viruses, 2009 pandemic, the American swine, the European swine, and the old seasonal. And you can see the American swine is really the outlier here. Very few cells infected. This is at 8 hours after infection, one full replication cycle, whereas lots of cells infected in the other two. And if we quantify that, we can see that by 12 hours after infection, the pandemic strain's almost taken over the culture. 75% of the cells are infected. And the American swine strain's really lagging, whereas the other strains have, by 12 hours, pretty much all passed this, this critical threshold. And part of why we think this is happening is that the neuraminidase activity, that activity for the virus to come off the cell, is much higher in the pandemic strains, and the Eurasian strain, which is what donated the neuraminidase to the pandemic strain, than it is in the American swine strain, which is otherwise the backbone of the pandemic strain, or of the prior seasonal. So this higher NA activity is going to allow the virus to spread through the culture and potentially spread through your lung more. Of course, it has to be balanced by the HA activity, but that apparently was optimized in the pandemic strain. And so these growth phenotypes explained why the pandemic virus was able to acquire a rapid growth phenotype compared to the European swine virus, the American swine virus, and is mediated by this neuraminidase, and I didn't show you the data, but also the M of the European swine virus. And we found that the European swine virus could transmit efficiently in ferrets as well. And so we started to try to model this. And these are similar to the models that you saw yesterday, where we're just looking at differential equation models. We're modeling infected cells that become latently infected, that then become productively infected, that can die at some rate. There's no immune response here. And then we're modeling the free virus that we measured. And this fits a lot of the data very well, but what it doesn't explain to us is why the European swine virus wasn't a pandemic. So the European swine virus had been going around for 70 years. We had never had a pandemic with it, and it grows just as efficiently in a cell culture as the pandemic strain virus. So we felt like we had to add in an element of the immune response, but we still just focused on the innate immune response rather than the adaptive immune response. And so we're measuring the innate immune response in the epithelial cells. And so this is looking at RNA gene expression of cells infected for 12, 16, or 24 hours with these different viruses. And what you can see is if you just focus on the 12-hour time point, which are these first two columns, everything in these two pandemic strains here are black. Similarly, with the American swine, also black, meaning that basically, even though the cells are infected, nothing's happening. It's as if the cell has failed to detect the virus is there. Whereas in the Eurasian swine and in the old seasonal, there's quite a lot of activity. So lots of genes have come on. Those pattern recognition receptors have recognized that the virus is there quickly. Whereas here in the pandemic strain, even though almost 75% of the culture was infected, no cells have noticed that the virus is there. And this is just putting that in really stark contrast. These are the top 8 genes turned on in the seasonal flu and the Eurasian swine. And these are all type 1 interferon response proteins. You can see all these I's that stands for interferon. So this classic antiviral response, and none of those responses are on in the pandemic strain viruses. And so the way we've started plotting that, which I find really useful, and I think is useful also in some of our SARS-CoV-2 studies, is we plot the amount of innate immune response we measure in these epithelial cells as a function of the total amount of virus genomes in the cell. And so each dot here is a different time point, different dose of virus, colored by the different virus strains. And the farther to the right and down you are on the graph, the more virus it takes to turn on the equivalent amount of innate immune response gene here. And so you can see the purple, the Italy strain, needs very little virus, very little viral genomes measured by the M gene here to turn on the same amount of innate response as the pandemic or the American swine strains needed to turn on that amount. And so it's almost a 2 or 3 log difference in viral genomes before the innate immune system pattern recognition receptors in the epithelial cells can recognize that the virus is there. And that's just summarized here for a number of different genes. We then spent a long time making recombinant viruses where we swapped different gene segments from the American swine lineages to the pandemic strain lineages to try to find what element of the viral genome of these 8 gene segments were potentially mediating this, what we call this stealthy virus effect in the pandemic strains. And to cut a lot of work short, no single virus gene segment could account for it, but collections of gene segments could account for it. And so these are spoiler alerts for both of these books. It was basically all of the gene segments optimized working together, really coming from that triple reassortant backbone that caused that massive evolutionary spike in swine that allowed the pandemic strain viruses, and that's represented here, to really distinguish themselves from the European strain viruses, which were able to turn on really high levels of innate immune responses with very low amounts of viral RNA. And so what we found then is that the pandemic strain is both efficient in terms of its replication and stealthy in terms of its ability to avoid the innate immune response. And that stealthy phenotype was mediated by contributions of the polymerase genes themselves with the non-structural genes and the proteins that coat the viral RNA itself. So it was all a package together that hid the viral RNA from those detectors inside the cell, those pattern recognition receptors. And so now we model that in our system by adding the innate immune response, and it's just as a general equation, not modeling any particular function of the innate immune response. And we fit that model by using specific gene readouts of innate immunity. And if we evaluate different models with and without the immune response and with and without cell regrowth after the cell dies from being infected with the virus, we do get, and we rank these models using something called the KK information criteria. Andreas will talk about that when we talk about fitting. But the key thing to know is that the lower the number, the better fit of the model. And our best fit model was this one with immune response, reducing virus production. So shutting down the virus production rate early on in the viral replication cycle and allowing cell regrowth. And we could account for most of the behavior of our system as a result of that model. And that just shows some of the fits there. Okay. And the last, I know I'm running into our question time, but these are going to be much shorter slides because we don't have modeling, but I just want to show you, compare and contrast how flu and really all RNA viruses share a lot of similarities, and the small differences in their pathogenesis really determine how they manifest in our population. So coronavirus is one of these positive sense RNA viruses, but it's single-stranded. It's a very large genome. It's almost 3 times the size of influenza, or twice the size of influenza. And it has, but similar concepts. It's an enveloped virus. It has something like the HA, but we call it spike. And spike does all of the work of the HA and the NA, basically an influenza virus. A similar replication cycle, except that spike in the case of coronaviruses often have a specific receptor. In the case of SARS-CoV-2, that is a specific protein called ACE2 that spike binds to very tightly, but it gets inside a vesicle. It has to get out of that vesicle, and then it has to go through its replication cycle, which for coronaviruses is actually also quite complicated because it's single-stranded, and it makes all of its subproteins off of this one single-stranded template genome. And none of this occurs inside the nucleus, versus in influenza virus, all of the replication occurs inside the nucleus. So different exposures for the innate immune response, but similarly replicating in respiratory epithelial cells, and needing to do that because it needs enzymes to modify the spike protein to be efficient for the next stage of replication. So it still has that cleavage event. This is a very common theme. Coronaviruses don't replicate inside the nucleus, as I said, but they do form these really weird big lipid globules inside the cell to sort of hide out from the innate immune detectors inside the cell and scaffold all of their replication machinery, whereas influenza, as I said, just sort of hides out in the nucleus. Already mentioned the distinct receptor binding capabilities, where ACE2 is a very specific receptor for SARS-CoV-2 versus the broad sialic acid residue of influenza. The coronavirus genome is much larger than flu, and as a result, it's able to spend a lot more of its real estate encoding several type 1 interferon antagonists. So that type 1 interferon response, it's a recurring theme, that this is what we use to fight viruses at epithelial surfaces and mucosal surfaces. And because of all of this complexity in the coronavirus genome and its sheer size, it makes a lot of proteins that just try to mess with that response. And so these are these non-structural proteins. Remember, flu had 1. Coronaviruses have 16, which is crazy. And we don't know what most of them do. So NSP3 as an interferon antagonist, NSP1 as well. Coronaviruses also have a number of accessory proteins that distinguish the different strains beyond SARS-CoV-2 as well. The coronavirus replication and transcription it has this incredibly large genome, and it basically makes these ever-increasing transcripts off of that genome, finally making the very largest gene products sort of in the lowest amounts. And this includes a lot of the replication machinery of the virus, because it sort of works from the end back up to the 5' end of the genome. Just summarizing that the SARS-CoV-2 includes all of these non-structural proteins that we don't fully understand what they do, but we think most of them are directed towards interrupting the innate immune response of the host cell, which is probably part of what allows SARS-CoV-2 to persist in cells much, much longer than influenza virus is able to. Protective immunity against SARS-CoV-2 is basically going to use the same mechanisms as influenza. And so I spent a long time on that, so I won't belabor it here, but we're going to have antibody responses that are going to block spike neutralizing antibodies from binding to the cell and letting the virus inside the cell. And then once that happens, if that blockage is not complete, we're going to need CD8 T cells to kill the target cell and finally clear the virus, particularly for this virus because it's hiding out in the cell and really antagonizing that intrinsic epithelial cell antiviral response mediated by the type 1 interferon pathway. So this just summarizes those similarities and differences. You can see a lot of the commonalities, and I just want to mention, because it's a virus of such incredible medical importance, and 3 more slides, RSV. So RSV is another negative sense virus, but so like flu in that sense, enveloped like flu and SARS-CoV-2, but single-stranded, so more like coronavirus in that sense. Has, again, it's surface proteins that's going to be used to attach to get inside the cell, and it segregates its attachment from its fusion. So remember, flu did that all in HA, and corona did that all with spike. And for RSV, it's going to attach with one protein and then fuse the membranes with another protein. This virus is by far the most important medical virus for infants. So the #1 cause of hospitalization for infants in the US, but even worldwide in terms of severe disease, is RSV. And we'll talk in 2 slides briefly about why that is and why it's different than flu. The viral replication cycle is, again, somewhat similar to flu. There's membrane fusion. You've got to dump the genome out, and you've got to go from negative sense to positive sense back to negative sense, and then reform the virions on the cell surface. Big difference is that this happens in the cytoplasm and not in the nucleus, so more like SARS-CoV-2 in that regard. And so why is RSV sort of different than flu? What distinguishes it? The immune mechanisms are going to be basically the same, but SARS-CoV-2 RSV has a slightly different tropism than flu does, and it infects ciliated cells and goblet cells and causes this goblet cell proliferation in specific areas of the lung that the combination of goblet cell proliferation, and goblet cells are going to be your major mucus producing cells, and death of ciliated cells nearby cause these large clusters of dead cells to form in your alveoli, coincident with the large production of mucus. And so those of you that are parents with young children, one of the ways you know you have an RSV infection is it's super snotty. When a flu infection, you're going to have a runny nose. But RSV is that sort of like never-ending parade of mucus coming out of the kid's nose. And if that mucus parade happens inside the lung, that's what causes the pathology, because it blocks the lung from performing gas exchange. And so it's this small difference in tropism that completely changes the pathological manifestation of RSV from flu to SARS-CoV-2, and specifically targets the reduced surface area of an infant compared to an older person, which will still get very snotty, but we have much bigger lungs. And so a little bit of mucus blockage is much less of a problem. It's worth mentioning SARS-CoV-2, worth mentioning RSV, because one of the reasons we don't have a vaccine for RSV is they tried to develop a vaccine for RSV on the model of the influenza vaccines of the 1960s. They gave it to a bunch of children in a trial, and 80% of those children who got the vaccine when they were eventually got RSV suffered very severe disease, more severe disease than the kids that didn't get the vaccine. And actually 2 of the kids died from getting RSV only when they had the vaccine. And so what it was determined happened there is exactly the same protective responses as we see in flu. So you either have, and SARS-CoV-2, you need antibodies or you need cellular responses. But the type of response that was mounted by the vaccine was not that type 1 response. For precise immunological reasons, what it generated was something more called a type 2 response. This is normally what we use to fight big pathogens like worms. And so because we had this type 2 response, we made sort of the wrong kind of antibodies and a reduced cellular response in these kids that had received the vaccine. And so when they were challenged with the virus, they were actually worse off than if they hadn't seen the vaccine, because if they just saw the virus, they would naturally make that type 1 response. And so this has been the sort of main sort of lodestar of having improved and more rational vaccine design, and a lot of careful preclinical trials now for any development of an RSV vaccine in particular, because we know a slight shift in the immune type that's generated can really make kids much more vulnerable. Okay, I'm going to stop there. There's one more question in there, which is, how does coronavirus leave the cell? Why doesn't it bind back on the cell? Why doesn't it need a neuraminidase? It's not really, I think, fully understood. It doesn't need a neuraminidase. It doesn't cleave spike off the cell. But what does happen is when coronavirus infects the cell, ACE2 gets internalized. And so cells that are infected with SARS-CoV-2 actually naturally don't have ACE2 on their surface anymore. And so as the virus tries to leave the cell, there's nothing going to be holding it back in place in the same way that sialic acids will be grabbing onto influenza virus.